Good afternoon. I'm Kilty Inafuku, and as Matt said, I worked this summer on chemical indicators for the rapid assessment of human fecal pollution in storm drains. There we go. So historically, Santa Barbara and other coastal cities have seen high levels of fecal contamination in their recreational waters during periods of dry weather. This pollution can come from both human and non-human sources, but we're most concerned with human because it has more potential to cause illness to humans and beachgoers. Recent studies have shown that this human fecal pollution can be found in storm drains, which is not good because that pollution gets washed untreated out to the ocean and exposes beachgoers and swimmers and surfers to harmful pathogens. And so, an overarching goal of this project, which is in collaboration with the City of Santa Barbara Creeks Division, is to identify the sources of that human fecal pollution in the storm drains. So my research is kind of a subset of that project, and we wanted to see if chemical indicators, or chemicals within the water, could be used to identify and screen for human fecal pollution. And so to do that, we measured the chemical indicators with the calorimetric method, and then we compared those results to what we got with a fecal indicator bacteria method and a DNA-based method specific to humans. So to overview the calorimetric method, we went out into the field and took a bunch of samples, and we measured the chemical indicators, or these 5 chemical indicators in those samples, and we chose these indicators because typically they have high levels in sewage, with the exception of nitrate nitrogen. But we decided to keep that with our other analytes because ammonia can be oxidized to nitrate nitrogen when it percolates through the soil, for instance. And the way we measured these chemical indicators is with this calorimeter right here. So what you do is you take your sample water, and you put in this little cuvette right here. You add a couple of reagents, and it changes color. And based on the intensity of that color, your device outputs your concentration. But before we could do that, we needed to check the accuracy of this device and check to see if there were matrix interferences in our water sample, which I'll explain later. And then moving on to microbiological methods, we wanted to measure the fecal indicator bacteria in our samples. This is a culture-based method currently used by the EPA to monitor water quality. And fecal indicator bacteria are just bacteria found in the intestinal tract. They're not harmful, but they're a good proxy, or they're supposed to be a good proxy for pathogens in the environment. And then we also have our DNA-based method specific to humans. And basically we extracted DNA and amplified that DNA with respect to the MNIF gene, which is a methanogen found in human pollution. And so if we saw that the MNIF gene was present in our sample, that meant there was human fecal pollution. So now onto our results. I said we need to check the accuracy of our calorimeter. And to do that, we created several calibration curves for each analyte, or each indicator. So shown here is the calibration curve for ammonia nitrogen on the left. And what we did is we had a solution of known concentration, and if we measured that concentration to be something different, that would tell us the accuracy of our device. So we had a known concentration, for example, of about 1.8 ppm, and we measured it to be 1.6. So even though it wasn't 100% correct, it was a very linear relationship for all of our concentrations. So whatever we measured, we can convert back to the actual concentration. And then we wanted to see if there were interferences in our sample water. Basically, we did that by performing performing standard additions for each chemical indicator. And basically, we went out into the field, collected a sample, and measured the concentration, which you see here in the blue. And then we spiked that sample with a known amount of chemical. So we added 1 ppm of ammonia nitrogen to our environmental sample, and that's what you see here in the red. What we expected to see is in the green. So we knew there were interferences in our samples if the percent difference between the red and the green, what we measured and what we expected to see, was over 10%. Most of our samples were under 10%, so there weren't many matrix interferences in our samples. And these are all the sites that we measured, we took samples from in Santa Barbara. So here you see the 101, and then here's the pier at the end of State Street. And we took a variety of samples because all these sources can contribute to storm drain water. So we had some creeks higher up in the mountains. We had some creeks lower in the watershed. We had a groundwater source. And the ocean samples, the green, don't contribute to the storm drains, but it was good to have as a reference. So these are all the concentrations we measured in our water samples. And you see a bunch of variability in our storm drain water samples, but what you should take note of is that in our pristine creek samples, you see some background levels of sulfate. So if the pristine creek is a contributing source to your storm drain samples, then it might not be the best indicator. Just because if you measure a concentration in your storm drain sample, it might be from the creek. And similarly with the groundwater, there were some elevated levels of nitrate nitrogen. So that might not be the best indicator either. And if we look at the individual chemical indicators in different types of samples, we see that nitrate nitrogen, phosphate, and the surfactants have potential to be good chemical indicators based on the fact that the levels in the storm drains, the blue diamonds, are much less than the values in sewage. And on the opposite end, sulfate and nitrate nitrogen might not be good indicators based on that same fact. Levels in the storm drain samples are much higher than those in the sewage. So if you measure something in your storm drain sample, you don't know that it's from the sewage. It could come from other sources. Then we went to our DNA-based methods. And this is that same map where we saw the presence of the MNIF gene, meaning we saw fecal contamination at these sample sites. And so if we did see presence of human fecal contamination, then ideally we would see a higher concentration of our chemical indicators. So like I said, nitrate, nitrogen, phosphate, and the surfactants were likely to be good or potential chemical indicators. And that's because they had higher concentrations where we saw human fecal contamination, at least for the really high concentrations. And when you get towards lower concentrations, it's kind of iffy. So we did have some false positives and false negatives. And I included nitrate nitrogen because, again, we saw a high concentration where there was human fecal contamination. But like I said, it might not be the best indicator just because it's not typically high in sewage. It just could be converted from the ammonia nitrogen. And these, the EC and ENT, that stands for E. coli and Enterococci, those are the fecal indicator of bacteria that we used. And so you can see that there aren't necessarily high concentrations where there was presence of human fecal contamination. And so these might not be the best indicators, even though the EPA does currently use it. So it's one reason we might want to look into using chemical indicators instead of fecal indicator bacteria. Conclusions from my research this summer. It is good to use standard curves and calibration curves when you're using calorimetry. Similarly, you want to check to see if there are matrix interferences in your sample water. And by matrix interferences, I just mean there are other chemicals in the water or other substances that could interfere with the quantification of your chemicals. And we also, as you saw with the map with the red dots, we did see human fecal contamination in several storm drains. So it's kind of important to know where that is. And the highest concentrations of our chemical indicators did indicate sewage pollution, so you could use those for where it is more highly contaminated. Future work, we want to analyze more samples just to kind of increase our bank of data. And instead of just checking to see if there's presence of human fecal pollution, we want to kind of quantify that. with a PCR assay. And we also want to possibly include other sewage indicators, such as caffeine or cotinine. So I'd like to thank the UC Leads program, Erica, Matt, my PI, Trish, she couldn't be here, and my mentor, Braun, who's in the back. And the Student Academic Preparation and Educational Partnerships, they give me funding, and the City of Santa Barbara Creeks Division. Thank you.